Thank you. Thank you so much. Good morning, everybody, and thank you for being here. Let me add my thanks to Peter Marks for the work that they're doing, but also taking Q&A in this. That was unexpected and a real pleasure. Let me introduce my colleagues from the gene therapy group. They represent Sarepta, Pfizer, Regenics Bio, and Solid, and welcome them onto the stage. And as they get up to talk, they'll introduce themselves, and then they'll get a bit of time to talk about each of their individual companies in that space. Do I have a clicker?
There is a clicker. Dan has it.
Oh, have you got a clicker?
There you go. There's your clicker.
Thank you. Okay, so I'm going to spend a few minutes really just introducing the concept of what is gene therapy. It means that the companies can then just jump straight in to start talking about their programs in some more detail, and that's what we'll do. We'll go through this here. So what is gene therapy? You know, it's fairly simple. It's been around for a while. I heard Peter talk about the number of products that's approved in the United States. But it's very simply that it allows us to fix or replace a defective gene and provide a real functionality into a cell. In Duchenne, the gene is large. We can't just simply put dystrophin back into a cell by injection. We use an AAV virus in order to carry the coding information for that gene. Viruses have evolved over millennia to be very efficient at getting their genetic material into it, and we've adopted that there as a way of actually being the basis of gene therapy. Multiple products are approved in this space. Lux Turner, I've given you an example for an eye indication, Zolgensma for SMA, Elevidys we know, and there's others. So I see this as a burgeoning field that's really going to expand over the next few years. The cartoon actually shows how nature has designed this and how we've adapted an AAV in order to be very efficient at getting genetic material inside a cell. It very efficiently gets in through the cytosol into the nucleus to deliver genetic material. And in our case, that genetic material is a copy of the dystrophin gene. The benefits of this are that it's a single dose payload. It's a high transduction rate. AAV has got broad focus for lots of different types of tissue, low immunogenicity and long-lasting therapeutic effects. But it comes with some challenges. Duchenne is one where we're trying to target all muscle. That's approximately 40% of body mass. And that means that we need a very high dose of an AAV in order to target all those different types of muscle groups. And that there has its own significant challenges associated with it. There's a limited cargo capacity. This is a very small virus. It's about 25 nanometers in diameter. And that there means that the actual carrying capacity is very limited, much smaller than the size of the dystrophin gene. And we actually have to truncate that sequence quite significantly in order to fit it inside a virus. And that there's part of the challenges that each one of the companies have faced up here. And I'll just walk through how they've managed to get around that. The full-length gene, the dystrophin full-length gene, is the largest coding gene that we've got in the human genome. And it encodes for a very large protein, a very complex biological protein. But the cargo carrying capacity of just under 5 kb really dictates what we can do in there. We have to severely truncate this gene in order to put it inside that. And nature has guided the thinking that we've had really to being able to truncate the gene down to a size that can fit into that. And that's really come from a number of observations. The cartoon that I've got at the top really shows the full-length dystrophin transcript, starting at the N terminus all the way through to the C terminus. And you can see the whole middle of it is a section of repeats. That their central rod domain is a focus for being able to truncate this gene. And that there really is based on key observations that we see in the real world. One of the most celebrated papers comes from a family of individuals that had a severely truncated form of dystrophin, which is shown in the bottom here. That their individual had exons 17 through 48 that were deleted, giving you a coding region of just over 6 kilobases. That they're still too large to put into an AAV, but that there became a template that scientists could work on to reduce even further. And what you see there is an individual who was still ambulant whenever he was in his 60s. He had a family member that in his 20s was a weightlifter. And so the take-home message here is you can severely truncate this gene and provide both very powerful phenotypic changes to almost an asymptomatic disease. And that there really has been the whole basis for which scientists have worked over the last decade to design various even shorter forms of this that can fit inside a virus. The gene therapy product is made up from several components. One of them is the capsid, one of them is the promoter, and the other one is the transgene. And as we look at each of the company's drug products, you're going to find that each of them are slightly different from each other. They're using different capsids, different transgenes that have been designed, and different muscle-specific promoters in there. Common features of them include the N-terminal actin binding domain. That there's a critical component for binding these microgenes inside the muscle cell. They've each got 2 to 3 of the 4 key hinge regions that are in the rod domain. They've got 4 to 5 of this 24 spectrin repeats that actually make up the middle of the dystrophin gene. And they've got a truncated C-terminus. All of this allowing them to get to a size that can be packaged inside the virus. Now, there's main differences between each of the 4 structures that are there. There's 4 individual AAV serotypes that have been used by the companies. Regenics Bio is using AAV8, Sarepta is using RH74, Pfizer is using AAV9, and Solid are using a brand new capsid that's been designed in their labs that has reduced the targeting for that, for liver, and improved the targeting for muscle. And we'll see how that plays out, but that's now in clinical trials, and we're going to hear about that in a few seconds. The 4 different muscle-specific promoters are being used all the way through. And you can see the ones that each of the companies got on the slide: SPC512, MHCK7, MCK, and CK8. The Regenics promoter is slightly smaller. It's been designed that way to be efficient and still target muscle and promote within muscle. But because it's smaller, it allows them to have a slightly more space inside the virus, and they've used that space to make a slightly larger transgene than the other companies. And you can see on the cartoon, they've got a little bit more of the C-terminus in there, and I think that there's going to be an interesting play out as that develops in clinical trials. I think I've said what I need in here. Oh, there's one more, that Solid has added a different sequence within the rod region to include a very important nNOS binding domain. So I think the take-home message here is there is 4 distinct products that each of the companies have designed, all individual from each other, all have got separate challenges within the science that have been resolved, and that gives us, I believe, 4 different shots on goal to see how each of these are going to play out. We've seen data right now, obviously, for Elevidys, is that their drug is approved. Pfizer have just completed the phase 3 Cifro study, and we're going to see top-line data this year, I think somewhere in the early second half of this year. Sarepta, obviously approved for 4 and 5-year-olds with Elevidys, and we're looking towards the FDA doing a review on that there over the next handful of months. Regenics Bio are in the Affinity study, recruiting in their phase 1/2, and have really released details of how they're planning to do their pivotal study later this year. And Solid are recruiting into their phase 1/2 study in what is the INSPIRE study. So you didn't come to listen to me. I think what I wanted to set up was that there is a number of really strong scientific thoughts that have gone into each of these drug products. They're different from each other, and I expect those differences may manifest themselves in clinical trials. So without further ado, I'm going to ask Dan Levy to introduce himself in the Pfizer program and talk about an update on that one, and we'll run down the line with Sarepta, with Regenics Bio, and then with Solid.
Thanks, Mike.
You're welcome.
Thank you. So I'm Dan Levy. I'm the team leader for gene therapy at Pfizer, for gene therapy for Duchenne muscular dystrophy at Pfizer. I have some positive things to share with you today, but also one very serious negative. As most of you know, we had a fatality in our Daylight trial, which was a trial in 2- and 3-year-old boys receiving fortidylstrogene. So before I show you a few slides that I was planning to share, I'd like to tell you what we know at this point and what we're doing. This boy was treated with fortidylstrogene movaparvovec 15 months ago. He was 3 years old at the time. And about 3 weeks ago, he collapsed. There was medical personnel nearby. They recognized he did not have a pulse, and he was treated immediately and appropriately for cardiac arrest, but he couldn't be revived. This is obviously tragic for the family and for the Duchenne community. At this point, we don't know if this was primarily a heart issue or if this was due to something else that caused the heart to stop. And we don't know if this was related or unrelated to the gene therapy. So there is an autopsy underway. And unfortunately, we've not been given any preliminary information from this autopsy. We've been told we need to wait for the final report. We hope that will come within the next month. But until then, we just have to wait. So what are we doing? The dosing in the daylight trial has already been completed for all of the 10 boys. The CIFRIO trial, which is the placebo-controlled randomized trial, phase 3 trial, there are still some boys who have not yet received their crossover dose. So, for example, some of the boys in the trial received a dose of either placebo or fortidistrogen a year ago and haven't yet received the crossover yet. And we've made a decision to pause dosing for now in that trial until we know more. So within the next month or so, we do expect to have those autopsy results and hopefully a better understanding of what caused his death. And we'll also have efficacy or effectiveness data from the CIFRIO trial at that time as well. So we're just finishing up collecting data from the last few participants we need for our analysis. And when we have that top-line report before the end of next month, we're going to look at the safety information that we have at that time and the efficacy information and hopefully be able to make a decision about resuming dosing in the CIFRIO trial. At that time, we'll also be speaking with regulators from around the world, including the FDA, about the appropriateness of submitting for approval for treatment with fortidistrogen based on the safety information we have and the efficacy. As just like we at Pfizer do, the regulators, their job is to look at the balance of safety and benefit. So it may be a little bit uncomfortable. It is a little bit uncomfortable for me to say it at this time, but we are actually rather enthusiastic about the efficacy results that we're going to see next month. We haven't seen anything. It's completely blinded, but I'm going to share with you a little bit of data that we have from our phase 1 trial. And I'll be showing you data that was presented a few months back at the MDA Muscular Dystrophy Association meeting, studying 16 ambulatory boys who received the same dose of fortidistrogen that we've been treating boys with in the daylight and the CIFRIO trial. Whoops, sorry. There we are. Okay, so this first slide here is a safety slide. And this slide should look actually very, very similar to a slide that I shared last year at the Cure Duchenne meeting in San Diego. So this slide covers safety information from the phase 1 trial, but let me just say that apart from the boy that I was just discussing, in the last year we've not seen any new kinds of safety events at all. So I'm now going to switch to our effectiveness data. And again, these are 16 boys receiving that same dose of fortidistrogen. But the phase 1 trial was an open label trial. And as most of you folks know, Duchenne is very variable in how boys change over time. And so it's rather difficult to just look at a handful of boys and say, oh, they got better. You really need something to compare them to. So what can we compare them to? We worked with an organization called CTAP, and CTAP is one of Cure Duchenne's partners. CTAP received their seed funding from Cure Duchenne. And they've collected natural history data from studies from around the world looking at DMD patients and have created a model where you can plug in information like their North Star scores, time to rise, 10-meter walk run, age, height, weight, and so on. And you can plug that in for an individual and make a prediction of how that boy may perform in 1, 2, or 3 years later. So we did that for each of the 16 boys in the trial. And what I'm showing here is how those boys compared to what would have been predicted in that model if they had not received fortidistogene. So when the bars go up, that means that they did better than the model would have predicted, and when they are down, it means they did worse. So the younger boys, 6 to 7-year-olds, are on the left, older boys on the right, and you can see there that the younger boys consistently did better than predicted. For the older boys, a bit of mixed results. Now, I'll note that the three boys that are represented on the extreme right side of the slide all had slower time to rise and slower 10-meter walk run times at the time that they received their dosing. And it's possible that they had more fibrosis in their legs and less muscle fibers able to be supported or protected by the gene therapy. But that's not to say that older boys won't necessarily benefit from a gene replacement therapy such as fortidistogene. We looked at their upper limb scores. So you folks have heard of the North Star score. There is an upper limb battery called the Performance of Upper Limb, or the PUL 2.0, that we used. And for the most part, across the board, we saw PUL scores that were above what one might expect for natural history. And we also we used MRI to look at muscle size, muscle bulk, and we saw a relationship between changes of their muscle size over this 3-year period and the results on either the North Star scores or the PUL tests. So let me show you the MRI results. So here now we're looking at MRI changes in the thigh, the lower limb. Again, younger boys on the left, older boys on the right. And the blue lines represent the percentage of increase over time of the muscle mass in the 16 boys in the trial, sort of split up again by age. And the red lines show MRI data that we collected in a separate study, in a placebo group in a separate study a few years back. So we see that the younger boys consistently and durably had an increase of their muscle mass, not so much for the older boys. And again, these are average scores, but the individuals, there was a relationship for each individual, whether if he had an increase of muscle mass, he was likely to have a relative improvement of his North Star scores. And so now we see the upper limb MRIs, and we see that in both younger and older boys, there was, on average, increases, and durably so, over these 3 years. And once again, the boys who had the most increases of their muscle mass in their arms tended to have the greatest increases of their PUL 2.0 scores. So, putting that together, in general, we're very excited about the possibility that we're going to see some strong benefit in the CIFREO trial. We'll know that soon. We also consider this to be very encouraging for older ambulatory boys. Once again, we're going to have additional safety and additional efficacy information in about a month's time. So with that, I want to thank Deborah and Cure Duchenne, and I'll just note that Fortidistogene movaparvovec was developed in the lab of Jude Samolsky, who was supported by Cure Duchenne. I also want to thank you folks for listening to me, and most importantly, we're just so grateful to the families who've been participating in our clinical trials. Thank you very much.
Thank you.
Hi everyone, I'm Robin Van Poelheist. I'm a medical director at Sarepta Therapeutics. So I want to thank you all for being here today. It really is great to see such a great turnout, and I really appreciate all the time and effort that you guys took to come here. So up front, I'd like to address a little bit of an elephant in the room. So we are in the middle of a data quiet period, which is a data review period with the FDA, and this is as we await to hear back from them about our potential label expansion for delandistrogene moxeparvovec. So my slides today are a bit limited. Our target review date is June 21st, and we will, of course, share updates after that when appropriate. So with that being said, again, it's great to see you all here today, and it's really great to see all the support from the Duchenne community. You can read this in more detail on our website, but essentially my talk does contain some forward-looking statements, and this does come with risks and uncertainties. So we do have a large clinical development program for our gene therapy for DMD, and what I'd like to do today is talk to you a little bit about the drug development process before we dive into our actual clinical trials and clinical development program. So what we start with is a proof of concept, and for a gene therapy, this can look something like designing and successfully showing that this gene therapy construct behaves as we expect it to in a cell culture dish. And from there, we will move on to animal studies. So this can be in mice or rats or non-human primates, which are monkeys. And as long as we're seeing the same positive effects that we're hoping to as expected, and we're not seeing that the risk outweigh the benefits, then we will move on to human clinical trials. And I'd like to point out that it's this early preclinical stage here that Sarepta partners with the patient groups and make sure that we get the patient group voices incorporated early and ensure that we have that continuing voice and input incorporated with our plans moving forward as well. So once we start clinical trials in humans, we're going to be focusing on safety in a smaller group of patients at the beginning of these trials with a smaller focus on efficacy. And then as we move to the later stage clinical trials, we're going to be focusing more on efficacy, but with a continued focus on safety. So it's at this stage when we have the clinical trials concluded that we're going to have a data analysis and FDA review period. And so this can actually take a very long time, just because the clinical trials are concluded. That doesn't mean necessarily that a drug will be available soon. So this can take a really variable amount of time depending on many factors. Once the FDA does approve the drug, however, and it is on the market, then we will have continued studies, post-marketing studies, and this will be to confirm and expand our understanding of the safety and efficacy of the drug. So as I mentioned before, the proof of concept is just the beginning, and this is a very long road. However, Sarepta does have studies that have covered all of these stages, and we currently have the only FDA-approved gene therapy drug on the market. So it can take many years and even decades for a gene therapy to move through this whole entire process, beginning from proof of concept and then going on to the post-marketing studies. And one thing I want to point out here also that is a little bit unique to gene therapy and what you've heard a lot about during the last talk as well is the importance of this manufacturing process. And for a drug to be commercially available, it has to have these processes in place. And for gene therapy, this is actually one of the most complex and resource-intensive parts of this whole process. And so this is different from what you would think about with the traditional medicines that come in the form of a pill. And another thing that's important to note here is the importance of post-marketing studies when it comes to accelerated approval, which is especially important with regard to our gene therapy, as that is the currently approved pathway that we've used. So this slide is a little busy, but it represents the body of work behind our clinical trials. And what I really want to highlight here is the number of years that we've invested and continue to invest for our DMD gene therapy program. And what I'd also like to highlight here is that this illustration only actually begins at the early clinical stage, so this actually doesn't even illustrate all of the years we've put in, but it is all of the clinical development program years. And another thing that I would like to highlight here is the depth and breadth of patients that have been included and are continuing to be included in our clinical trials. So to date, we have dosed over 200 patients in our clinical trials, and this includes patients over a wide span of ages and genetic mutations, as well as ambulatory and non-ambulatory patients. So for example, in our studies 302 and 303, we have a very wide age range, going all the way down to newborn and going all the way up to no age limit on the non-ambulatory patients, respectively. We're also doing what we can to overcome known issues. So for example, in our 104 study, we are looking at a way to overcome pre-existing immunity to our gene therapy, to our construct that carries the gene therapy, which is our vector. And so this would be to be able to treat patients that have pre-existing immunity to our vector or high antibody titers. We also have our sights set on long-term data collection and monitoring. So in our 305 and 401 studies, we're looking long-term at patients that have been in our clinical trials and then commercially treated patients, respectively. So we are doing all that we can to reach the largest number of patients in the community possible. This thing is sensitive. So we would love to learn more from you guys and stay connected. If you have questions or comments, please connect with us at advocacy@sarepta.com. You can also check out Duchenne.com for more information on Duchenne, as well as drug development processes and pipeline. And then we also invite you to join us on social media. We would love the chance to get to know everyone better. So thank you again to Cure Duchenne, and I especially want to thank Cure Duchenne for the early financial support that they provided to Sarepta, as well as their ongoing support through the patient advocacy group efforts, as well as in our direct partnerships. And especially thank you to all the patients and families in our clinical trials.
Well, thank you so much for all of you having us here today. My name is Naz Dastgir. I'm the Senior Medical Director at Regenics Bio. I'm also the clinical development lead on our RGX-202 program, which is a Duchenne gene therapy. And I'm also a pediatric neuromuscular specialist by training, so my heart's really in this work. We're so happy to be here today. So I'm going to talk to you about RGX-202, which is an investigational gene therapy for the treatment of Duchenne. And I'm going to share a little bit of our interim clinical data just to hopefully get you a little bit excited like we are. Here are our obligatory forward-looking statements, so I'll move on to the next slide. So here's some information about the Duchenne gene. I know that Mike had spoken about this earlier. So our transgene is designed to encode the key elements of naturally occurring dystrophin, So the normal dystrophin is up there at the top. You can see how many bits and pieces it has included in it. And we have the dystrophin from a Becker phenotype patient that shows that there's actually a lot missing that can lead to a lot of better function. And the RGX microdystrophin, a lot of the other microdystrophins and the transgenes that are designed are trying to get as much of that Becker phenotype gene packaged into that small vector that was mentioned before, trying to deliver as much bang for your buck as you can get in one vector delivery. And so we at Regenics have developed RGX-202 as a new differentiated microdystrophin with the important biology that is most similar to the natural shortened dystrophin that protects muscle from degenerating. So I know you saw this on Mike's slide earlier. So RGX-202 is novel among the current classes of AAV microdystrophins. There's a huge list of these here. Ours is up at the top. We use an AAV8 vector, an SPC512 promoter. I know Mike also mentioned the hinges. We also include hinge 3. That can be a difference among different transgenes. And we also have uniquely the C-terminal domain. And that C-terminal domain binds to some other proteins in the sarcolemma or the muscle membrane, trying its best to create as much stability to the sarcolemma so that there's less degeneration of the muscle or breakdown over time. So we currently have a study open and enrolling. It's known as Affinity Duchenne. We are enrolling children, boys between the ages of 4 and 11. You must have genetically confirmed Duchenne muscular dystrophy. At present, we're only enrolling those with mutations in exons 18 and above, and that's mainly due to concerns about safety that were raised in the past, but hopefully that will change in the future. We require the kiddos to be able to walk about 100 meters without assistive devices, and you need to have no pre-existing antibodies, and that's because it could render the drug ineffective if you have pre-existing antibodies to our AAV8 vector, or sometimes increase your risk of toxicity with that drug. So this is what we've done so far as part of our study. So we've done some dose finding studies where we're trying to find out exactly when we know what's effective in mice, but could you do higher doses, or could you stay at a lower dose, or what's the best and safest dose? So based on mouse studies, we found out that a higher dose is better, but a low dose is also effective. So first we start off with a low dose, the lowest effective dose. We dose 3 children with this. It's 1 × 10⁻⁴ genome copies per kilogram. And we got a safety approval, not just internally, but from an external review monitoring committee. We then went on to dose level 2, which is 2 × 10⁻⁴ genome copies per kilogram. We actually dosed 2 patients here, got an external monitoring committee to say, okay, this is looking safe and good. You can move on to an expansion cohort. And we're currently in the middle of dosing our expansion cohort. So the green check marks mean that that group is fully enrolled. So now we're just on that last phase here to try to fill up our expansion cohort. And obviously it takes time to see what the functional data looks like, but we do have some information from our muscle biopsies that I'll share with you later that basically show how much of our microdystrophin gets to the muscle tissue itself. And hopefully that's a good surrogate biomarker, a measure of that your drug is getting to the right place and it's hopefully producing the benefit that you're looking for functionally. So this is our timeline of assessments. So we have some screening assessments that take place early on. And then during that screening period, even before you get dosed with the RGX-202, we actually have an immune suppression regimen that's a little bit more involved than some of the other studies you've heard about. We use prednisone and sirolimus and eculizumab. And some people raise questions about that, what is the need for it? Well, thankfully we have had no serious adverse events in our study, and we really do attribute it not only to the safety of our drug because it has some unique features to make it more safe from an anti-transgene response, but also from the perspective of the vector response that you can sometimes see that can lead to some of the adverse events that you see with gene therapy. So we've kept going on with it. And it's very well tolerated. So the children will get the immune suppression regimen. They get dosed with RGX-202 on day 1, and then have regular follow-up. You also, during the screening period, have a baseline biopsy before any intervention takes place, and then 12 weeks afterwards, you get a follow-up biopsy. And then we also get the functional data that goes on through month 12, and then we also do some MRI studies as well of the skeletal muscle and the cardiac muscle. So here are some of the key baseline characteristics and safety data that we've seen. So as I mentioned before, from a safety standpoint, we've had no serious adverse events. We have had some common AEs that are just like nausea, vomiting, but obviously I'll just kind of go through what doses that we have here. So our first dose is, so the dose here, 1, 2, 2, is dose level 1, dose level 2, dose level 2. We had the dose evaluation cohort or group of patients. We dosed 3 patients between 4.4 to 10.5 years of age, weighing 17.8 to 28.3 kilograms, and we have 7.9 to 13.4 months of follow-up, doing well. In terms of a safety standpoint, we don't yet have public information on our functional data, but hopefully we'll be able to share that with you soon. For dose level 2, we had, so there's the dose evaluation phase and the dose expansion phase, but those patients ranged from, I can give you the details for the whole group there, so 5.8 to 12.1, weighing between 17.3 to 31.2 kilograms. And we've had follow-up for those patients for up to about 5.9 months. And what we've seen is that with dose level 1 and those 3 patients who were dosed, where we were trying to decide if we're going to go with dose level 1 or dose level 2, those patients had between 11.1 to 83.4 microdystrophin expression at 12 weeks after dosing. We also saw a reduction in CK levels or creatine kinase levels at 10 weeks after dosing, and you know CK is it's often how you get the diagnosis of Duchenne, is that your CK is elevated. So we're monitoring that closely to see if there's any correlation with that. For dose level 2, we have data from biopsies on 2 patients. We have a microdystrophin expression at 12 weeks ranging between 20.9 to 75.7%. So we're very excited about these results. We know it's reassuring us that the drug is getting to the right place. It's hopefully supporting the sarcolemma like we'd like to see it, and we're hoping that it's going to translate well into functional data as well. This is just a summary slide of exactly what I just told you, and in the interest of time, I'll move on to the next one. This is just a sample slide that shows something called immunohistochemistry, which is an antibody stain for your protein product. So we have an antibody stain for dystrophin, but it stains the portions of our microdystrophin that are captured within the dystrophin staining. So you can see the normal on the far left, where all of those muscle fibers are rimmed with red. The baseline in our patient has no rimming at all. And at 3 months, we can see that that microdystrophin is rimming the muscles and getting to the right place. Because you know, when you have your vector and you have your promoter, the promoter is like, all right, I'm going to get you to the muscle. Well, let's make sure it actually gets to the muscle, and this is helping us understand, and the microdystrophin percentages as well, that it's actually getting to the muscle and going to the right place. So we do have a video here, but I don't know if we have time to share it. This is some of our patients, but it's fine, I'll move on, because we have 1 minute left. So all of our patients have been really great. They've been seeing positive benefit from like a home video standpoint. And again, we have functional data that's coming out, so hopefully we'll be able to share it with you shortly. And I'll give you kind of a summary slide here. So as of May 3rd, RGX-202 has been well tolerated at both dose levels with no serious adverse events. RGX-202 is a microdystrophin. RGX-202 microdystrophin expression was observed at both dose levels in all ages. There are encouraging observations of early improvements in daily activities associated with strength and function in clinic and caregiver videos. And Regenics Bio is now enrolling patients in an expedited dose level 2 expansion phase. The initiation of the pivotal phase is expected in late quarter 3 of this year, which is like the 3rd portion of this year, and early Q4 as we call it. And Regenics plans to use RGX-202 as a surrogate, microdystrophin as a surrogate endpoint, likely to predict clinical benefit. And just know that we have a really great team. Our heart's with you. We're very dedicated to the Duchenne community, and we're very grateful to be a part of this meeting. And we are so grateful to the families who have volunteered to be in our study. We are forever indebted to them. We're also very grateful for all the investigators that are involved with our study, all of the study coordinators, our team at Regenics, and of course, Cure Duchenne. Thank you so much.
All right. First of all, a huge thank you to Deborah, Mike, Liana, and the entire Cure Duchenne team, both for this amazing meeting and the opportunity to present. My name is Pat Gonzalez. I'm the Senior Director of Clinical Science at Solid BioSciences and the clinical lead for our SGT-003 program, which I will be describing today. So SGT-003 is a next-generation microdystrophin gene therapy. Mike and everyone here has done a great job in going through what is the gene therapy and the nuances of different approaches. And so here, as we'll go through our presentation today, we'll really go through what are the pieces that we have been motivated to move this next-generation product forward with. We are a public company, so our lovely forward-looking statement slide, apologies. And so as an overview for SGT-003 and the pieces that are key to this approach, we have three really main components that are driven by our novel capsid that is now our new delivery mechanism for this. One, as we went through before, there are different pieces of microdystrophins that can be chosen to be put into an AAV because you do need to have a shorter sequence that can fit into that capsid. And so ours is a unique transgene that does include an nNOS binding domain. With that, we are being delivered by a novel capsid that has been developed in the lab to get more of the product to the muscle, so being able to get a higher response and potentially do that with a lower dose as well has been our motivation to put our efforts into this program. We do that really also understanding that we need to make enough product to get to patients at these high doses, and we need to make sure that that's a pure product. And so we focus our efforts on a transient transfection manufacturing process where we've optimized the amount of full particles and true full particles so that we can both hopefully use this to improve on the overall safety profile, and we've also seen that improves the overall potency of it as well, so the effectiveness of the drug when we use this process. So as I mentioned, our microdystrophin has this nNOS binding domain. In non-clinical studies, this has been shown to prevent muscle damage that can be a result of ischemia. And essentially, when you use your muscle more, you need blood flow to get to it. nNOS and then the production of nitric oxide can allow that to happen. So this is a piece of the dystrophin that if we can restore, we're hoping for that to have additional functionality as well. And really based on the key research from Jeff Chamberlain, Dongsheng Duan, we've been motivated to continue to use this product as a specific microdystrophin that we think could have that added benefit. And so AAV-SLB101, this new capsid that we're using as that next generation gene therapy real focus, is a variant of AAV9 that was developed in the lab as a way to be able to get more to the muscle. And what we've seen in these studies, and here highlighting a mouse study, is that we can see multiple fold higher amounts of that vector get into quadriceps and heart, so important tissues for this disease. And at the same time, as we see more go to muscle, we see less going to the liver, so about half of the amount getting to the liver. And so these could be meaningful differences both in terms of the efficacy profile as well as hopefully for the safety profile as well. And when we compare SLB101 to first generation capsids, so the capsids that are used in the approaches right now, like RE74 here specifically, or AAV9 or AAV8, what we do see, and this was an NHP study, so a monkey study that was done where we looked at microdystrophin expression in the heart and quadriceps, that we did have multiple fold higher levels of microdystrophin expression in these tissues using our capsid specifically. So very exciting data for us to move this program forward. And so last year, the update we were able to say was that we were really putting together a package to get into the FDA and be able to move that into the clinic. I'm excited to present now that data that we've been able to generate to be able to put that package together, and then the details of our active study as well. So what you can see here is we initiated with a dose-ranging study to look at the amount of microdystrophin expression. This is a range of 3E13, then 1E14, and 3E14. So wanting to see, can we see that dose response? And interestingly, at all of these doses tested, by 1 month, we see 100% positive fibers by immunofluorescence staining here. That was sustained out as well. But so this was exciting data for us, knowing that in first generation capsids, especially AAV9, that we've compared a lot of this data to, we see that at these dose levels, we don't get these same responses. So we know we're seeing multiple fold differences by this measurement. That then translated as well to efficacy in terms of muscle function. What we could see is at all of those doses tested, we saw that the muscle function in these animals also was restored near wild type levels. So again, at very low doses, seeing a robust microdystrophin response that then translated to a functional response. Looking at biomarkers as well to be able to see, not just from an isolated muscle, can you also see circulating changes? Looking on the left side here, something that's very meaningful to all of us, which is circulating serum CK. We know that's increased in Duchenne, and here in the mouse model, the same is true. When we have our treatment here, even at that lowest dose tested, we have a very significant reduction that's near those wild type levels. So exciting data that really go together with our functional results. And looking at additional biomarkers here that we're evaluating, serum titin and AST as well, seeing those reductions similarly. And so because our first study really had such robust responses, we wanted to even go at lower doses and evaluate when do we start to see more of a dose response. And when we looked at 5 to 10 times lower doses, this is really with that now when we started to see that efficacy profile even decrease. So now going into the E12 range, so if we think about current dose levels, especially as we were initiating and looking at AAV9 studies with a 2E14 efficacious dose level, looking at 6E12 here as the first where we see significant differences in mice, it could be a potentially very meaningful difference. And then as we mentioned before with our microdystrophin, we have the ability to not only have a functional microdystrophin in that it's localizing where it needs to go, so here a dose response study seeing more microdystrophin that's at the muscle membrane as we increase that dose, but we're also able to restore nNOS to the membrane as well. So showing that that extra domain is adding that extra function. And so we were excited to say that this really, this package was put together and we were cleared by the FDA to move into the clinic. These data got us very excited, and so we have initiated what we are calling the Inspire Duchenne study. This is a phase 1/2 open label, non-randomized study. All participants do receive SGT-003. We are using a single dose level in the study of 1E14. And I want to point that out for a second because the results that I just showed were excitingly saying that in the E13 range and even lower, we see meaningful results back at in the functional efficacy. However, because we know this is a single dose treatment, we want to really focus on that dose being able to have that benefit that we need in our boys and men. And so with that, we chose the 1E14, knowing that there are gaps in first generation products that we could be able to overcome with the next generation approach that could add that benefit, but we want to have that meaningful effect. In this study, we are using immunomodulation that's focused on prednisone, so just around the period of dosing, and we're enrolling patients who are aged 4 to 7 years old. Right now, we have 2 active and enrolling clinical sites with excellent investigators and teams. Kevin Flanagan at Nationwide Children's and Perry Shea at UCLA, and these teams are working tirelessly really to get everything moving in this study. I'll go through a couple of the key criteria and the study details as well. So overall, I mentioned the ages that we're enrolling are beginning with 4 to 7 year olds. We're starting at a younger age because this is a new program. We want to primarily evaluate safety, which is ultimately the focus, and then from there, looking at strategies to be able to expand out as well. In this study, we do have mutation exclusion criteria that are related to that DMD mutation because of the potential for having an immune response against the transgene. So because our microdystrophins are a little bit different, our specific criteria are a little bit different as well. And we're beginning, again, very conservatively and then hoping to learn with the field to see how we can expand this as well. These criteria are any deletion mutations, so specifically deletion mutations in exons 1 to 11 or 42 to 45. Because these are boys here, we're also enrolling ambulatory boys, and these we do need to test, importantly, for the presence of antibodies against the capsid, and so that will be something that is done during screening. We also require that these boys are on a stable daily steroid regimen for 12 weeks so that we have some limitation of the variability and the consistency that we can get in this first cohort. Similar to most of these studies, because this is first in human, we do want to focus on safety, and so we're doing a robust safety assessment within those first 30 days, frequent visits really to make sure that we're checking up on our boys in the study, looking at lab values and making sure that everything is going well. From there, because it is a gene therapy study, we will have longer term visits, both looking at efficacy and safety as well. On the efficacy side, we will be performing muscle biopsies to evaluate importantly, our microdystrophin expression, 3 total biopsies within this study that we're aiming to be done by needle biopsy technique, and this is used at screening, day 90, and at day 360 post-dosing. From there, we'll also have functional assessments done to evaluate the efficacy in addition. And so we're really excited about this overall program. We want to be able to see, as we move a program forward with the next generation capsid, really what is this showing? It will be one of the first, if not the first, muscle-tropic next generation capsid going into the clinic. So hoping to generate a lot of exciting data over this year. And please, if you have any questions, feel free to come discuss with us. We'll have a table in the exhibit booth as well, and always come and talk to any of us individually. Our information for our 2 clinical sites are listed here on this slide, nationwide as well as UCLA. And for additional information, you can scan that QR code or go to clinicaltrials.gov as well. And so I want to thank you all for your attention, and thank, importantly, obviously, the entire patient community for just all of the efforts to be able to be a part of this research process and move this forward. Thanks so much.
Perfect. Thank you. We're right at the top of the hour, but I want to take a minute just to wrap up what we've heard right now. I started off by talking about there's a number of different approaches in here, different capsids, different transgenes, different stages of development. We have a drug approved. We've got one where we're going to see phase 3 data in a handful of months. Others are slightly earlier stages of development. And waiting in the wings is Insmed, waiting to come in and begin their phase 1/2 trials, I think, later next year. This is a really exciting time, and I think that we've got multiple shots on goals with different companies with different drug products in here. So an exciting time for gene therapy. I'll wrap up with saying that tomorrow we've got another gene therapy session to discuss some other components of this in detail. We're going to be talking about the latest learnings that we've got from the safety and immune consequences of this. I want to talk a little bit and expand upon new capsids, the way to deal with neutralizing antibodies. So there's a lot more that's going to come out and be teased beyond what the companies are talking about right now. And I think this is going to be a really exciting year, both with the introduction of Insmed coming in, but these continuing advances of these programs with new updates that are going to happen. So the final thing for me to say is, I really appreciate each of my colleagues coming along, and giving us all updates. I really want to thank them for all that they're doing in this disease, and also all of the patients that are participating in these studies. We couldn't get anywhere without that involvement. So thank you all of you, and thank you all of you. Okay, we're done.
All right, wow. How about that for a great start for this conference? 20 years ago, I would have never had a clue what they were saying. Gene therapy is very impressive. Thank you for the companies that were up here, and especially Mike. Thank you for laying the land so it's more understandable for me. In fact, Mike leads our team of Cure Duchenne scientists, Liana and Potin, and I think best scientific team in all of Duchenne, so I'm grateful for them. All right, it's time for a little break. As your Chief Fund Officer and Custodial Service Officer, CFO and CSO, those are not the way I should be responded to. You got 15 minutes. Take advantage of meeting some people, take a break. I told you where the bathrooms were earlier, and we'll see you back here for champions, the champion families, to hear about how Actions Therapy. Please come back. It's very impactful what can be done. So see you in 15.